Okay, so we're going to start out with the carrying of CO2 through the body. And there's a key concept here that happens across the membrane of the red blood cell called the chloride shift. Okay, and so the chloride shift is happening in our red blood cells. Okay, we talked a little bit about that bicarbonate reaction last time. Okay, and this is really allowing this to happen in red blood cells over and over and over and over again. Okay, without any buildup of the products of the reaction. Okay, so when the big thing here that we call the chloride shift is that we exchange chloride for bicarbonate in the red blood cell. Okay, the reason we need to do this, number one, is so we get no change in membrane potential. And really kind of probably before that, to understand it would be we need to get the products. Man, I think I need a new Apple Pencil. It's been doing that this whole week or last week too. Get products out. That says products. Sorry about that. To get the products of the reaction out of the cell. And so that one really kind of comes first, right? If we want to continue this reaction to keep happening in the red blood cell, which is going to be how we deal with CO2, then we need to get all the products of that reaction out, or it'll stop, right? When we build up the products of a reaction, that reaction will stop or it'll reverse. So we need to get those products out. But when we do that, that would be changing membrane potential of the cell potentially. So instead of allowing that to happen, we bring in a negatively charged ion and we're sending out a positive or a negatively charged ion as well. So those two things kind of go together. Okay. And really the direction of that exchange depends on where the red blood cell is. When the red blood cell is down at our tissues, which is where it's going to be picking up CO2, then bicarbonate is going to go out of the cell and chloride is going to come in. And then when we need to get rid of that CO2 so we can breathe it out in the lungs, which is happening down here at the lungs, then the shift happens the other direction. We bring that bicarbonate back in, we send that chloride back out, and then we can create CO2 again and release it in the lungs. Okay. The membrane protein that's going to help us do this exchange, just called an anion exchanger, anion exchanger 1. One thing that's kind of interesting about this is our venous blood has about 3% higher hematocrit due to the red blood cells swelling with water. Okay, the reason for that is because we keep that negatively charged ion inside the red blood cell, which creates some osmotic pressure, which is going to pull that water with it. Okay, and so yeah, that's going to cause some swelling of the red blood cell. Which is going to make the red blood cells bigger, so they're just going to take up more of the space of the blood. And then we also lose some water after like we talked about last time, after the blood passes through capillaries. So the venous side of our bloodstream has higher hematocrit than the arterial side because of these two reasons. Okay, so I think this will all make a little bit more sense when we look at these two images here. Okay. So, and I want you guys to kind of walk through these with the person next to you. So we got a red blood cell there. We can see some cells here. Okay. And then the next slide, we can also see another different space here, the red blood cell. We can see the reaction happening. Okay. There's a couple things I want you to do. Pretty much you can just kind of walk through these questions with the person next to you. So where in the body is this? The options are really just the tissues of the lungs. Okay. Carbonic anhydrase is a enzyme that's going to help drive this reaction. And then you can kind of see here what happens to that hydrogen ion that we create, because that could potentially be problematic. What happens with the bicarbonate? And then CO2, we have 3 different ways that we carry CO2 in the body. Okay. Number 1 is just in the form of bicarbonate. Number 2 is just dissolving CO2 directly into the blood. And then number 3 is CO2 binding to hemoglobin itself. Okay. We can see all 3 of those things here. Okay. So kind of just try to walk through the steps here that you're seeing and see if you can answer the questions on these 2 slides. And we'll come back together here in a sec. What does it do? Yeah, you can kind of see where it goes there on that slide. See it?
Hemoglobin is stabilizing.
Yeah, it's just like any and most proteins are buffers, right? And so it's kind of hemoglobin is acting as a buffer here by just allowing the hydrogen to bind to it. Yeah.
Thank you.
Mhm.
Where is that?
Good. How are you?
Okay.
Still happening.
You do have the snow jacket today. I was wondering when you
Yeah.
came in, you didn't have it. Yeah. It does get cold quick. I know. I wish there was some happy medium. Yeah, that's tough.
Like back in August, this room was like seriously like 35 degrees.
Oh, it was hot.
And I was like with a lady, and somebody was like, she was like, and like it was like really like I don't know
Yeah,
or something.
yeah.
But sure enough, we came in here on the first day and it was
Hey, see, it was working. Working well. All right, let's go ahead and come back together here. Let me just explain this one again. I don't think I explained that one very well. So venous blood, so thinking about the blood on the venous side after it's already passed through the capillaries, that whole side of our vascular system compared to the arterial side before it gets to the capillaries has a little bit higher hematocrit because of the fact that red blood cells are either going to have chloride inside of them or bicarbonate. Only on the venous side though, because this whole system is due to picking up CO2 at the tissues, right? And so on the other side, we haven't done that yet. We haven't picked up CO2, so we don't have the chloride or the bicarbonate inside. So it's just adding some osmotic pressure to the cell, which is going to pull some water with it too, right? So then our red blood cells get bigger, so they take up more of the space of the blood, right? That's all hematocrit is just what volume of the blood is made up by red blood cells compared to plasma. And so that increases it quite a bit. Right. So that makes sense. Somewhat. Yeah.
it increases the osmotic pressure.
Inside the cell, and then that pulls fluid in, which makes the cells swell a bit.
What hematocrit is?
Hematocrit is just the percentage of our blood that's made up of red blood cells. And so since they're getting a little bit bigger, that's why that percentage goes up. Yeah. Okay. All right. Okay, so then let's go back to this here. Okay, so where in the body is this one? Tissues, yeah, because we're making CO2 releasing it, right? We're seeing CO2 come out of those tissues that are doing metabolism, sending it into the blood. So we're picking up CO2 here, right? Okay. So carbonic anhydrase works by helping put CO2 and water together. Okay. So it's really going to be driving this reaction here. Okay. And that only happens in our red blood cell. We don't have carbonic anhydrase just floating around in our blood, but we do have that enzyme inside of a red blood cell. So that's the biggest reason that this arrow, these arrows, the size of these arrows represent how much of that reaction is happening. Because we can do that reaction out here in the blood too, right? But not as much because we don't have that enzyme helping drive that reaction, right? So it's just a lot faster when we have carbonic anhydrase helping us out. Okay, so that's where it's happening. It's inside the red blood cell. Okay, and so one of the potential problems of this, so obviously we're getting CO2 in the cell there, right? CO2 comes in, it combines with water via carbonic anhydrase, turns into carbonic acid for just a second, right? And then it can dissociate into bicarbonate and hydrogen. The hydrogen could potentially be a problem, right? We build up a bunch of hydrogen, this could be an issue, but hemoglobin is a buffer, right? Most of our proteins are buffers, right? So that hemoglobin can just bind right to, or sorry, the hydrogen can just bind right to the hemoglobin, and then it holds on to it. And then look what happens too. This is exactly what we were talking about last time, that shifting of the curve, right? High hydrogen ion concentration shifts the curve to the right, which makes us drop off oxygen. This is part of what's doing that, right? The hydrogens binding to the hemoglobin make it so that the hemoglobin doesn't have as good of a strong of a hold on oxygen. So this helps us kick off the oxygen as well, right? So it's kind of all working together. Does that make sense? Yeah, so we see that. So we see buffered by hemoglobin. Okay, with the bicarbonate, it's going to be sent out of the red blood cell. And this is the exchange for chloride. Okay. And then again, the reason we have to do that is because if we just built up a bunch of bicarbonate inside the cell, then this reaction would stop, right? We wouldn't continually be able to bring in CO2 and convert it into bicarbonate. And then CO2 would build up in the cells, and then we would lose our diffusion gradient for CO2 as well. And then the red blood cells couldn't pick up CO2 anymore, right? So we have to continually kind of move this along to make that reaction be able to be continue happening. Okay, so obviously the chloride comes into the cell, right? Okay, and why does it need to leave the cell? Kind of answered that on the two previous slides. Number one, we need to get the products of the reaction out, and then number two, by exchanging it for the ion of the same charge, this maintains membrane potential and doesn't mess with that, which could change the structure of membrane proteins, all kinds of different things, right? If we're messing with membrane potential of a cell. Okay, so the three methods of carrying CO2. This is one, right? In the form of bicarbonate is happening here, both out of the cell and into the cell. This one is just purely dissolving CO2 into the plasma, which happens a bit. And then this one here is just combining CO2 with hemoglobin, which we can do as well. And that also helps to push oxygen off, right? That's part of shifting the curve as well, is adding CO2 to hemoglobin. Okay, questions on that first one? Go ahead.
Sorry, can you repeat the middle, the nest arrow that's happening?
This one, is it this one? Yeah, it's the same reaction. It's just happening in the plasma instead of in the red blood cell. Yeah, we just don't have carbonic anhydrase out there in the plasma to help us with that. So it's just slower and we don't do as much of it that way. Yeah, and then, yeah. The top one here? Yeah, it's just dissolved, like any gas can dissolve in water. It's the same idea. There's not much of it there. I don't think I forgot to do that. On the previous set of notes, I had the percentages. It's something I don't have them off the top of my head, but 70, I know it's 70% happens here. Oh, you know what? I think I do have it in my notes. Oh yeah, 70% is in the form of bicarbonate. All right, so as far as those three methods go, 70%. It's in the form of bicarbonate. Okay, and then it's 20% via hemoglobin. And then the other 10% is dissolved in the plasma. So. Okay. Any questions on that, or we good there? Okay. All right. So that's at the tissues, right? So now this same red blood cell is going to make its way through our venous system, go through the right side of the heart, right? Get pumped over to the lungs, right? And that's where we find it here on this slide, right? So now it is in one of those pulmonary capillaries, right? And we got an alveoli or an alveolus right there. Okay. So this is obviously in the lungs. Okay, carbonic anhydrase is still happening inside the red blood cell, working inside the red blood cell. Okay, that's going to be happening to break this down, right? To help us break down that carbonic acid. And now we see that same hydrogen that bound to hemoglobin before can come off. We can use that same hydrogen, right? So this just comes off, and this is due to the fact that there's a big push for oxygen to come in, right? There's a high concentration of oxygen outside the cell, so it's going to push inside the cell, which helps to kick that hydrogen off of the hemoglobin, right? And so that's going to help kick the hydrogen off. The hydrogen is going to go up and then be able to bind with bicarbonate here that came back in via this chloride exchange, right? Chloride left the cell now, bicarbonate came back in, it meets up with the hydrogen that was waiting for it that came off the hemoglobin, goes into carbonic acid for just a second, right here, right? Carbonic anhydrase breaks that down, turns it into CO2 and water. CO2 goes into the lungs and we can breathe it out. Okay, and all these other methods are the same, just reversed. Okay, go ahead, Lisa. Yeah. Yeah, it's just like reversing the, it's just, yeah, just a reversible reaction that enzyme drives. Yeah. Yeah. Other questions? Yeah.
Just for the HCO3, is it coming into the cell to then leave the cell again as CO2?
Exactly. Yeah. Other questions? Okay. We good? Yeah, go ahead, Yvonne. Yeah, it can stay in the red blood cell. It can just kind of, you can go out of the red blood cell. It's just kind of there's so much water everywhere in our body that it just kind of equalizes depending on osmotic pressure where it needs to go. Some of it's going to go into the alveoli too, right? Some, because we're going to breathe some water out every time we breathe. And so it just kind of, is everywhere. So, Okay. Other questions? Yeah, go ahead.
Sorry, but the protons, they're still buffering, but do they have a higher concentration near the tissues? But is that, I guess that's external though, because that's from the tissues.
Yeah, are you asking why it comes off of hemoglobin here?
What happens to it?
Oh, what happens to it? It binds to bicarbonate. So it comes off of hemoglobin right here, right? So it was attached right here. We got this hydrogen plus hemoglobin, and then it comes off. Now we got hemoglobin. So the oxygen kind of helped kick it off, right? And now we got hemoglobin with the oxygen bound to it and the free hydrogen. So then it's free to then go up and bind to bicarbonate. So it has a lot to do with the concentration of oxygen, that if there's enough of it, then it's going to actually have a stronger affinity for the hemoglobin than the hydrogen did, right? So then it can kind of help kick it off. And yeah, that makes sense. Yeah. Okay. Other questions? Okay. All right. So that is CO2 transport. Let's go ahead and talk a little bit about the acid-base balance in general, and then we'll talk about how the lungs help out with this. It will be a little bit of review from our very first class back in that August? I guess it was August. Yeah, we talked about that a little bit. That was a while ago. Yeah. Like, whoa, that was a long time ago. Yeah. Okay, so acid-base balance. A few reasons why it matters. Enzyme function. right? All of our proteins are pH sensitive, right? And all of our enzymes are proteins. Almost all of our reactions need enzymes in one way or another. And so pH is very important for that, right? We can have proteins denaturing, right? That can be similar to what we're just talking about with enzyme function, but could also be like structural proteins as well, right? And ones that aren't, or membrane receptors or anything like that that's a protein could be affected by pH, right? And then ion solubility. So things like our bone can break down if our pH gets too low, right? Other ions that have become solidified in different ways will start dissolving. And so this can happen with long-term metabolic acidosis. Our bone, we can lose bone mass, things like that. So those are just 3 reasons why this is so important for us. Okay. So we got a few major buffer systems in the body, right? The purpose of these is just to prevent large changes. Prevent large changes in pH, right? We're allowed to have small fluctuations either direction, and then that buffering system kind of helps us pull that back. Okay, but all buffering systems have these 2 components, a weak acid and a weak base, right? We're just looking at that with the bicarbonate one, okay? And then our 3 major buffer types in the body, proteins, like we were just talking about with hemoglobin. Plasma proteins do a lot of this, okay? Because we have that free carboxyl side and then that amino or the base side of our amino acids. Okay, hemoglobin, very, very powerful compared to our normal plasma proteins. Okay, you guys might understand what this means more than I do. Histidine residues, something like that. You guys know that from biochem? Yeah, okay, what does that mean? You want to tell me? Okay, yeah, all right. Okay. Thank you. Perfect. Yeah. All right. Yeah. The reason why this is so powerful is just because we have so many of them, right? We have so many 250 to 280 million hemoglobin in a single red blood cell, right? And then we have 25 trillion of these red blood cells, like we talked about last time, right? So extremely powerful buffering by hemoglobin. And then this one is one we just talked about, our number 3 there, carbonic acid bicarbonate buffering system. Okay, so acid-base balance, we'll just kind of walk through this briefly here. Okay, CO2 and hydrogen, obviously we're just looking at this, or CO2 and water, turn it into this, right? So if pH increases, maybe I'll have you guys just do this briefly with each other. So if we have a disruption of homeostasis where pH is increasing, which way does this equation need to move to help us fix that? And then same thing if pH decreases. Okay, just review that with the person next to you for a second, see where we're at, and answer those little questions underneath each. Come back together in a sec.
You're gonna blow it out. Oh, sorry. I'm thinking about. Yeah, you're right. What? You're a mean part.
You gotta send her back over there. Yeah. Okay. Did you guys do this in biochem as well, like as far as like buffering systems? That something you guys talk about, or in? Okay, just hemoglobin.
No, we didn't.
Okay. All right. Okay, so let's come back together here for a sec. So in our blood, we've got all these components all the time, right? Those things are always there. It's not like we just have the bicarbonate side or just have the acid side. We've got it all, right? That's the kind of the key of this buffering system. We've got the weak acid, we've got the weak base. So which one is the weak acid here? Should have started with that question, maybe. Now you guys know this. You guys teaching me chemistry up here, and then okay, all right. Huh? Sonella, what did you say? The one on the right. Thanks for glad you tried. That's the acid, right? That's the acid. Okay. Bicarb is the base, right? This is just a hydrogen ion. Okay. So yeah, carbonic acid is H2CO3, right? That's a weak acid. And this just kind of shows you nicely what a weak acid means. If we take a strong acid and put it in solution, it all dissociates, right? It's all of those hydrogen ions are going to be donated. If we put a weak acid and put it into a solute, take a weak acid, put it into a solution, some of those are going to dissociate, some of them aren't, depending on the already concentration of hydrogen ions there. If there's already a bunch of hydrogen ions in that solution, then not many of them are going to be released. If there's not many at all, then it's going to release more, right? And that's kind of the idea here. Okay, so we have this solution with all these things in it, which would be our blood. Okay, if all of a sudden our pH increases, what does that mean for our hydrogen ion concentration? Does it go up or down? pH increases, that means hydrogen's what? Down. Okay, so we got a decrease in hydrogen ions. Okay, so which one of the two components of this buffering system do we need to help us out with that problem? The acid or the base? The acid's the one that has the ability to add hydrogen ions to a solution. Okay, so the acid will dissociate. Okay, so it'll add hydrogen ions to the solution. Okay, so what does that look like as far as the equation goes? Does it move to the right or to the left? To the right. All that means is this one turns into these two, right? That's what it does. If we just take H plus and remove it from H2CO3, then now we got one H, and now it's negative instead of neutral, right? So that's all we're doing. We're just moving that equation to the right. Okay, so then pH is going to go which way? Up or down? Down. Good. Okay. Questions on that? Good. Okay, so then if pH decreases, if we have a disruption the other direction, what does that mean for our hydrogen ion concentration? Up or down? Up. Okay, so we got more hydrogen ions. Okay, and so then the buffering system, which one do we need to help us out with this problem? Base. Okay, so the HCO3 negative, that bicarb can accept hydrogen ions. Okay, and then that equation is going to go the opposite way. It's going to go to the left, and this moves our pH back up. So this is our primary buffering system as far as like chemical buffering system of the blood, of the extracellular space. And then we'll see the phosphate buffering system is the primary one that helps us out inside of cells. But okay, questions on this at all? Okay, so then, oops, other way. Yeah, so then we'll kind of walk through these different acidosis and alkalosis problems that we've talked about. This is the thing I was saying is going to be reviewed from that first class. We talked about these on the first day. So the definition of acidosis is a pH of 7.35 or lower. Okay. And if we're talking about respiratory acidosis, what is it that's causing this? Ask each other real quick. So give me a thumbs up for breathing too much or a thumbs down for not breathing enough. Got it. Got it. So hypoventilation. right? We're not getting rid of enough CO2. So then that pushes that bicarbonate reaction that we're just looking at back the other direction, right? And we end up making hydrogen because that's what's on the far other side of that reaction, right? Is some hydrogen, okay? So any short-term rise in the partial pressure of CO2 above 40 millimeters of mercury can cause this, okay? Emphysema is part of this. We get so much air trapped in our lungs, right? That residual volume goes up so much, okay? Narcotics, right? Where we're slowing down our breathing rate, right? We're just not getting enough breathing going on. That can build up CO2. And so then there's two ways that the body compensates for this. Really only one, because it's really only the kidneys that can help out with this, but the kidneys do two things for us, which we'll talk about those in the next couple of weeks. But we increase our release of hydrogen via the kidneys, and then we keep more bicarbonate in the kidneys. And remember, the reason we don't say that breathing is a compensatory factor here is because breathing's already the problem, right? Of course, we're trying to get them to breathe more, but that's just not like the immediate way the body responds because breathing's already the issue, okay? All right, so that's respiratory acidosis, okay? Respiratory alkalosis. So this is going to be a pH of 7.45 or higher. Which ventilation will we put there? Hyperventilation. Right, we're blowing off too much CO2. Okay, so any short-term decrease in partial pressure of CO2 below 35 millimeters of mercury, anxiety, being at high altitude, panic attacks, things like that could definitely do this, okay? Compensation is just going to be the opposite. So we increase reabsorption of hydrogen in the kidneys. We're holding on to more hydrogen, and then we're decreasing the retention of bicarbonate. We could say we're increasing secretion of bicarbonate. That would really mean the same thing. And then we're retaining more hydrogen. We'll get into more of those terms next time when we talk about the kidneys, but put in more hydrogen, holding on to more hydrogen, putting more bicarb in the urine. Go ahead. That answers your question? Okay. All right. Okay. So that's our two respiratory ones. Okay. And now the metabolic ones. Okay. So metabolic acidosis. This is that we're making too many hydrogen ions for some reason. Okay. So this could be ketosis, like we've talked about. All right. Diabetes mellitus, could be starvation, which causes ketosis. Okay. Both due to liver forming ketone bodies. Or it could also be severe diarrhea. That feels like this bullet point needs to be pushed back a level. Not part of starvation. Yeah, so severe diarrhea, we're losing the bicarbonate rich juices from the pancreas, right? From the pancreatic secretions, bicarbonate rich, we're losing a lot of those. So now we can talk about both the lungs and the kidneys helping us correct for this, okay? So we're going to increase ventilation to blow off that excess CO2. And then we're going to also get rid of hydrogen via the kidneys and hold on to more bicarbonate in the kidneys. Okay, so when we have these excess hydrogen ions, that is going to make us breathe more, right? Blow off some of that CO2, and some of that breathing, that excess breathing that we see is this Kussmaul breathing that we see from when people are in ketosis. Okay. All right, so metabolic acidosis, and then lastly, metabolic alkalosis, right? So we're losing hydrogen ions for some reason, okay? Certain diuretics can cause this, okay? Because they're causing us to shed a bunch of water. Sometimes hydrogen goes with that, depending on the drug that we're talking about. We'll talk about some of those diuretics in the next 2 weeks. Vomiting can cause this because we're losing that hydrogen-rich stomach acid, right? And then the compensation is just the opposite. So we increase the reabsorption of hydrogen in the kidneys, decrease retention of bicarbonate, and then we slow down our ventilation, right? We're trying to hold on to CO2 via not breathing as much. Okay, questions on those? All right, so this next one is a nice little image called the acid-base nomogram. When you first look at this, it feels a bit daunting, okay? But if you can kind of analyze this in somewhat of a similar fashion to like the Wiggers diagram, looks pretty daunting when you first look at it. But then once you get it, it's like, wow, that really helps understand the mechanics of the heart. This to me feels somewhat similar to that. Okay. So let me just kind of set it up for you a little bit. And then I want you guys to look at it a little bit and try to interpret some of it. Okay. So number one, those red lines, this is like a graph that has like 5 axes. So that's why it's a little bit tricky. Okay. The red lines are representing the partial pressure of CO2 or the concentration of CO2. Okay. So for example, right here, you have to follow this line all the way up to get your partial pressure of CO2. Right. So anywhere this line is, the partial pressure of CO2 is 70. Okay, that makes sense. So it's kind of like not an x-axis, not a y-axis, not a z-axis, some other weird axis, but that's what it is. Okay. And then we've got the x-axis is just the arterial blood pH. Okay. Y-axis is the plasma bicarbonate concentration. I don't know what you call that top axis, what letter that would be, but the top here is the blood hydrogen ion concentration, which is obviously going to go very well with the arterial. So it's probably just like the secondary x-axis, something like that. And then which I believe you call the z-axis over here, this is our, that is still the CO2 thing, right? It just kind of wraps around, right? So like when this line hits right here, 20, right? So it's kind of like this CO2 wraps around both sides there. Okay, so that's just kind of setting it up. Staying in that like pink part or purple part, whatever you call that, you can kind of see where metabolic alkalosis is, chronic respiratory acidosis. You can see all the different ones. You can see normal right in the middle. Okay, so here's a few questions I want you to look at and try to interpret. Okay, number one, so what is the difference in chemical concentration of blood between chronic and acute respiratory acidosis? Okay, so we start with acute respiratory acidosis right here. This would be like when it first starts happening. And then here's what it looks like when we've given it some time, but given our body some time to adjust to that. Okay, so chronic means that's lasting for, maybe a day or longer, and your body has made some adjustments to that. Okay, so that's what I want you to first interpret there. Same thing here. Okay, what do you think is actually causing those changes? Okay, and then these two questions as well. What's the difference in CO2 levels between respiratory and metabolic acidosis? And then what's the difference in CO2 levels between respiratory and metabolic alkalosis? Okay, so see if you can kind of come up with some answers on that based on interpreting that graph. And then we'll come back together in a few minutes. Okay, go ahead.
Enough for because it's a scale.
Or is it the difference?
For which one?
For any of them, like if I.
Yeah, just pick a spot.
Okay.
Kind of maybe somewhere around where the name of it is,
Okay.
Yeah, like the label.
So for acute respiratory acidosis, it doesn't have to be in the pink. It can be
Yeah, you do want to do it. That one's kind of weird because it's not there. Yeah, choose somewhere in the pink. Choose somewhere in this pink. Choose somewhere. Yeah, choose somewhere in the pink part. Yeah, good question. Yeah.
What are we?
So just like, what's the difference in hydrogen ion concentration? What's the difference in CO2 concentration? So you're kind of just like trying to see if you can bicarbonate. You're seeing what kind of differences you see there. Yeah.
For chronic. Yeah.
I'm thinking, and then maybe if it's acute, I haven't been able to like.
Everything goes up a little bit more.
Sorry. Gotcha.
Yeah, I got anatomy. The written portion come back.
Okay, gotcha. Yeah, I got you.
Yeah. Yeah, I didn't realize there's three alphabets. Respiratory, yes. I don't. Thank you.
All right, let's go ahead and come back together. It's like a lot of you are diving into this, trying to figure it out. So let's start with the acute versus chronic respiratory acidosis first. So acute respiratory acidosis would be here, right? But really kind of in this part here. And then chronic, we make our way up this way. Does anybody want to offer something that they noticed there? What's kind of a difference there? What do we say? Chronic has greater, which one? So that one, I mean, it depends on where you're looking. But if we just like went from here to here, the CO2 stays somewhat the same, right? Because we're following those red lines. But it could be a little bit because this one kind of ends here, right? Whereas this one kind of stretches out. So that when we're increasing CO2, it would be, it'd have to go this way, right? For the most part, we're kind of going this way as we're going from acute to chronic. So we don't have a huge change in CO2. Yeah, I mean, you're kind of, yeah, it's definitely not, you know, we're kind of just looking for trends mostly, right? It's not because we don't have a specific numbers, right? So we're kind of looking for trends. And so in general, we're heading this direction, right? And that's the exact direction that the CO2 lines head. So it does kind of keep CO2 stays somewhat similar there. Yeah, Jeremy, what'd you think? More bicarb floating around for sure, right? Because we're going up here, right? We're going this direction, which is up on this scale. Okay. And why would that be? Did you guys talk about why that would be, Jeremy? Yeah, and this right here, we're talking about respiratory acidosis, right? These are our two ways we compensate, right? We hold on to more bicarbonate, right? So that's what we're seeing. This is showing the compensatory effect of the kidneys right there, right? What it means by chronic, it means first of all, the kidneys take a while to cause their compensation for pH, whereas the lungs are fast, right? We can breathe out a lot of CO2 fast, but with the kidneys, it takes a while to urinate out things, right? We're not urinating all day, right? Like we are breathing. And so the thing we're seeing here is we're seeing that increase in bicarbonate. Okay. What did our hydrogen ion concentration do? Just shifting from here up to here is moving pH up a little bit, right? And it's moving hydrogen ion concentration down a little bit, right? Which is what we would hope if we're trying to compensate for a low pH, then it's going to have to, hydrogen ions are going to have to go down, right? So both of these are the effects of the kidneys doing their compensation that we just talked about that they would do for for respiratory acidosis. CO2 is going to be similar. And a lot of that has to do with the fact that we don't really change our breathing, right? If breathing is still slow, then we're still going to have, for the most part, like I said, it could, if anything, it could increase a little bit, right? If this, because this line kind of goes up this direction more than this one does, could increase a little bit, but for the most part, CO2 is going to be somewhat similar. We're seeing the effects of the kidneys compensated by going from acute to chronic. Questions on that? Does that make sense? Okay. Yeah, and I'm kind of already answering these questions. This is kind of answering that question, right? The kidneys compensated that. Yeah, Madison.
Sorry, just more of a general question.
Yeah.
For that map, the purple, I don't know how to ask this question.
Yeah,
Is
area.
it-
We kind of
I
want to.
guess, like for the acute respiratory acidosis, is it that whole purple line that's going out to the side, or is it just where it says?
It's the purple part. Yeah,
So from normal to the left.
yeah.
it's like what
That's
would
kind of the general area you would see these values
Okay.
for acute respiratory acidosis. Yeah, yeah. I know that you're kind of maybe looking like where the letters are. Yeah, I know that makes it tricky. I should have said that to start.
Like for the normal,
Uh-huh.
is it also just like where it says normal, that's normal? Or is there like a little couple squares that would?
Yeah, I mean, it's probably this whole area in the middle here, right? Something kind of like that.
But then when it's shooting off, that whole area is then considered the issue.
Yeah, exactly. Yeah. No, that's good. I should have defined that a little bit more clearly before we started. Thanks for asking that. Yeah. Okay, other questions on that? And then this one's kind of weird because it even kind of gets cut off by the word right there, right? It's kind of like,
But that, so
like,
that would go down farther.
I think it goes down a little further than that, yeah, but it's even kind of because it's so much shorter than the other one. But yeah.
The acute respiratory alkalosis would be that whole little section below it.
Yeah, the whole purple section down there, yeah. So, and then on the other side of things, acute respiratory alkalosis, right? So we're talking about over here, okay? That compared to chronic respiratory alkalosis here, it's a similar situation, right? We're pretty much following those CO2 lines straight down for the most part, right? So CO2 is going to stay somewhat similar, but just the opposite of what we just said, our bicarb is going to go down, right? And our hydrogen ion concentration goes up, or pH goes down, right? So it's CO2 equals similar, decrease in the bicarb, increase in the hydrogen. All right, again, that's caused by the kidneys doing their compensatory help. Okay. Questions on that? You're saying because this seems to be like a little bit closer. Yeah. Yeah, it's a good question. I think it's maybe because we're kind of on the low end of bicarb already at that point. Like we're already kind of down, this dropped just a little bit, right? But we're kind of nearing like real low levels of bicarb. And so yeah, I don't know if that explains it, that we just don't have as much room to move down there. Or if it has to do with the fact that it looks like also the pH disruption might be not quite as great as well. If we look like going from normal to here, I guess, that would be pretty extreme, like 7.6. Yeah, I don't know the exact answer to that, but yeah, I think it just has something to do with probably our normal range that we can move bicarb levels, but that'd be my guess, but I don't know for sure. Yeah. Yeah, it's a good question. Okay, and then let's look at CO2 levels here. Okay, So what we see as far as a difference in CO2 levels between respiratory and metabolic acidosis? Anybody want to just note what they saw here? Okay, so metabolic, let's look at them. Talking about metabolic acidosis, which is here, yeah, versus acute or chronic acidosis, respiratory acidosis here. It's a lot lower, right? Yeah, so we got a lot lower CO2. And why? Did you guys talk about why? What did you-
Would it be because you can now use respiration to compensate?
Exactly, right? We're seeing the compensation of the lungs by that, right? The lungs are releasing that CO2 to try to help us with this problem of hydrogen, right? And so we're seeing that, right? And that one happens quick. We don't need to look at necessarily the chronic versus acute, because our lungs adapt super rapidly. We can blow out extra CO2 really fast. Okay, so metabolic acidosis is going to have a decreased CO2. It's going to be due to increased ventilation to compensate. Right, and then it's just going to be the opposite here, right? So if we look at respiratory versus metabolic alkalosis, right, we see alkalosis up here, alkalosis down here, right? So that big shift, much higher CO2, right? That is going to be due to decreased ventilation to compensate. Right, so we can kind of see all four ways that the kidneys and the lungs are responding here in these different types of pH disruptions through this nice little graph that somebody who was really smart all put in one thing. I'm not sure how they came up with that, but can be kind of helpful. So questions on that at all? Okay. Yeah, let's just do like a 5-minute bathroom break. And then we'll come back, talk a little bit about hypoxia, and keep going. Okay. 5-minute bathroom break. Let's just come back at 10:40. All right, we ready? All right, let's go ahead and get started. We're going to talk a little bit about hypoxia here. So, yeah. Okay, so we got 4 main categories of hypoxia. Hypoxia just means low oxygen in the blood for some reason. Okay, so yeah, hypoxemia would be just arterial O2 is low. Okay, which is also just known as hypoxic hypoxemia. Okay, anemic hypoxia means that we have low hemoglobin for some reason. Ischemic hypoxia, also known as stagnant hypoxia, is we have low blood flow to a certain tissue. So low okay, so there's hypoxia in a certain regional area or local area because we're not getting enough blood flow to that area. And then histotoxic hypoxia is a certain toxin impairs O2 usage in our cells. Okay, so some toxin that might interfere with mitochondrial activity, disrupt mitochondrial enzymes, some sort. Okay, so all of these are obviously related to the use or delivery of oxygen in one way or another. So just different definitions of things that we can call hypoxia. Okay, so we'll talk about a couple in a little bit more detail. Okay, so hypoxemia due to high altitude, right? This is leading to low arterial oxygen saturation. Okay, so anybody know what mountain this is? What was that? What'd you say? No? You got it. Yeah, that's Mount Whitney for sure. Yeah, it has those good job. Have you hiked it? Yeah, when you go through Lone Pine, you see it right there, right? Yep, exactly. Yeah, it's unique because it has those three peaks like that. But highest mountain in the 48 contiguous United States. Did you hike it? Go to the top? No.
I just stopped by the
McDonald's, yeah. You can hike Mount Whitney or go to McDonald's, either one. Similar idea. That's great. Yeah, it is totally. And I know what you're talking about. Yeah, for sure. Yeah, it's a cool mountain. Yeah. Okay. So this doesn't. I have hiked Mount Whitney. Yeah. 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 Monique and I did it. It is 14,600 and something. Yeah, so it's the highest outside of Mount McKinley and stuff in Alaska. It's the highest in the 48 lower, higher than all the ones in Colorado, surprisingly. But very close. Some of the Colorado ones are like 14,400 and stuff. But yeah. So. I've driven higher than that when we were in Ecuador. Yeah, when we were in Ecuador last year. You can drive up to 16,000 feet. Yeah, yeah. I went to the highest mountain in Ecuador called Chimborazo, which is over 20,000 feet. And you can. Yeah, we drove to like 15, and then me and Soren hiked up another 1,000 feet to get to the snow. But it's
1,000 feet at that elevation.
yeah, it's right at the equator too. So the snow level doesn't even start till 16,000 feet. So it's the closest place on Earth to the sun, which is one of the cool thing about it. Because it's on the equator, right? And then it's 20,000 feet tall. So yeah. Did I wear what? Yes, you definitely need sunscreen up there. Yeah, thank you. Yeah. What's that? Oh, no, it's not hot. Yeah. But very strong sun still for sure. Yeah. 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 But we digress. Okay. So let's walk through kind of what happens when you go to a high altitude like that. So the big problem is a decrease in, man, this pen is killing me. O2, jeez, it's like barely working at all. O2 saturation, right? So a decrease in oxygen saturation is what happens just immediately because of the partial pressure of oxygen around your red blood cells is going to decrease. Okay. And so that causes you to start breathing more, right? That's just a direct stimulus to start breathing more. That increases your ventilation. Right, so the Y is that, right? That decreased O2 saturation. Okay, but if you're not actually increasing your metabolism, then this is a form of hyperventilation, right? Because you don't actually have extra CO2 to blow off. So since you're breathing more, your CO2 levels go down. Okay, which also is, which way would that change your pH? Up, yeah, hydrogen down, pH up, right? Yeah, so there, okay. And so if we've got low CO2, low hydrogen ion concentration, what kind of a shift in the curve do we get there? Left or right? Yes, we get a leftward shift of the curve. I guess I already have that there, sorry. And so this just helps us to actually load more oxygen, right? So this increases oxygen loading, right? So if we're at a situation like this, like right at 40, right, we would go from about 80% saturated to about 95% saturated, right? So this is a helpful thing that happens to your body when you first go to altitude. Helps you get some more oxygen on your red blood cells. Okay, this is also going to stimulate erythropoietin release when we get that low oxygen saturation up here, right? And that's going to eventually increase our hematocrit, but that takes a while. That takes a couple months to really kick in. Okay, questions on that at all? Okay. Yeah.
So when it seems like it's harder to unload at the tissues,
Yeah.
would you then like cramp more or like have issues with like ischemia because you're not?
Yeah,
eating?
it's probably not as big of an issue because the partial pressure of oxygen around our capillaries, like in the interstitial space, is also going to be lower too, right? And then you might have a buildup of CO2 there as well because of the extra metabolism you're doing. So it might affect it a little bit, but for the most part, since you've got a greater diffusion gradient as well, then it's probably not as big of an issue. Yeah, it's a good question. Okay. Yeah, other questions on that? Okay, so then another form of hypoxemia is caused by right to left shunting, right? We talked about the PDA already with the presentation, but there's three other defects of the heart here that would also cause right to left shunting. Right, so atrial septal defect, right? That's one that we were already talking about as well. That was on the exam as well, right? Just that going right from the right side of the heart to the left side of the heart and via the atria. But then we got these two others we haven't necessarily talked about yet. Atrioventricular septal defect, AVSD. Okay, this is really no septum at all, right? You're just seeing this big space here. There's just kind of this one common valve. It's in a way like a two chamber heart, right? And so this requires major surgery for survival, and survival rates are not super high with this defect. And then we've got ventricular septal defect is just on the ventricle side, right? So just down here, right? So there's a hole in the septum between the two ventricles, right? With AVSD, there's no septum at all, right? It's really just no separation between the atria or the ventricles. With ventricular septal defect, there's a hole in the septum between the ventricles. All of these obviously cause right to left shunting. Skipping of the lungs is pretty much the problem here, right? A lot of deoxygenated blood can just go right back out the aorta. And so instead of going over the lungs to get filled up. So, okay. Questions about any of those? Sure, you'll learn a lot more about those and some of the treatments when you get to your cardiac block. Okay. All right. So one more thing here related to hypoxemia is how does this connect to the VQ mismatch stuff we were talking about last time? Okay. So we look at kind of two such situations here. This would be kind of the left side is ideal, kind of everything is working right. We got good ventilation in the alveoli, right? We got good and even flow of blood to those alveolar capillaries, right? And then we look over to the uncompensated side. This is where we have non-uniform ventilation. Okay, so you can see good flow into this alveoli and then some blockage here. This could be a chronic bronchitis case, right? We have buildup of mucus in some of those bronchioles, not allowing for full ventilation into a portion of the lungs, right? And so this just kind of gives you some numbers here. Looking at alveolar ventilation, should be about 2 liters per minute in each A and B, which gives you 4 total, right? You can see the blood flow. And then we shift over here to this one, we can see that since this one dropped, since B dropped down to 0.8 liters per minute, this one's able to compensate for that a little bit, right? So we can see that extra ventilation going into the other side, which is the way our lungs will work. If part of them isn't working, we can kind of shift more air into the other parts that are working well, okay? But the blood flow doesn't adapt with this, right? So we still see the same blood flow going to each side, right? So we're still sending a lot of blood to this alveoli, even though it's actually not taking in as much air, right? So that ends up becoming the problem here, where we do end up with the same ventilation to blood flow ratio here with this 0.8, because you're adding the two sides together. But the problem becomes the saturation, right? So the blood going through this side gets saturated at 98%. But the blood going through the decreased ventilation side only gets up to about 92%, right? So now we're kind of combining those two. We have a little bit of loss of saturation there, right? And it's because, yeah, the lungs can compensate as far as sending more air to the ones that it's really just a pressure thing, right? If we're finding resistance in one part of the lungs, more of the air is going to go the other direction, right? But the blood flow is going to be similar, right? We're sending blood to alveoli that don't have as much oxygen in them, and so that blood is not going to get filled up as much as the blood that goes to the fully ventilated side of our lungs, right? So that's the big thing that I just wanted to point out there. Questions on that at all? Yeah.
Just further down, it's talking about how there's like increased arterial tension, at least in the second one, and the alveolar tension.
Yeah, so we're going from here to here. Yeah, so alveolar O2 tension, right? Yeah, so that's really a matter of like concentration difference of oxygen inside versus to the blood, right?
But is that an actual like pressure change too? So then like if more and more were blocked, is that what kind of causes pulmonary hypertension and like pressure to build up on the right side of the heart?
Hmm.
From that.
No, it's a different type of tension there. Yeah, because that is specifically looking at like concentration difference of the gases, not so much blood pressure. Yeah, great question though, for sure. Yeah. Other questions on that? Okay, so let's take a few minutes and have you guys go ahead and kind of work through these slides just to kind of recap what we just got done talking about. And then yeah, and then we will come back together and have yeah, I feel like we just had a break, but do we want to do another break? Let's do this first and then we'll see where we're at, okay? Should I pose next to it? Felt like everybody's taking my picture. Okay. All right. Oh, let me put the foot back on. Okay, we good?
Yeah.
Kind of Wilder? Yeah, good. All right. Okay. All right. So next we're going to talk about how we control ventilation. Okay. And so the primary ways that ventilation is controlled. So I think I've mentioned this, but just to reiterate these terms, hypernia is increased ventilation in response to a true increase in oxygen demand. This would be due to increases in metabolism. Okay, but then hyperventilation, as we've already been talking about, hyperventilation is a pathological increase in ventilation not triggered by oxygen demand. All right, go ahead.
Is
Yeah.
hypernia the same as like tachypnea?
No, hypernia is it's just our normal response to increased breathing. Yeah, it's normal. Hypernia is totally normal. It's what happens when we start exercising. Yeah. Whereas hyperventilation is an increase in ventilation that doesn't match our O2 demand. So then that's going to lead to that decrease in CO2 that we've talked about. And we'll be talking about in a sec, just how important that CO2 levels are for driving ventilation, right? It's the primary driver of ventilation. And so this is why hyperventilation can make you pass out because of those low CO2 levels. But okay, so a few things as far as the regulation goes. Okay, so voluntary control of our breathing is going to be happening via, obviously, in the cerebral cortex. Okay, but we have respiratory, and then those are going to communicate with neurons from the cerebral cortex are communicating with respiratory motor neurons in the spinal cord. SC for spinal cord there. And that's pretty much what we're seeing here, right? So higher brain control center, cerebral cortex is the voluntary part, right? Going to make their way through the medulla, but then eventually down to the spinal cord, down to those respiratory muscles. Okay, and then we have automatic control via a few different places here. Okay, so there's kind of two main parts, the cervical portion and then thoracic portion, and then this pre-Botzinger complex. Okay, so the cervical portion is what controls the diaphragm. Which nerve is that controls the diaphragm? You guys know that? Okay, and then the thoracic portion is going to be what controls our intercostal muscles. Both external and internal. Yeah, and those are mostly controlled by the intercostal nerves here. Okay, and so in the as far as the automatic control portion goes, we have actually pacemaker cells. in the medulla. Okay, and that's pretty much what we find in this pre-Botzinger complex. So these are similar to to the pacemaker cells that we found in the digestive tract, in the heart as well, that are depolarizing on their own at a certain rate to maintain a certain rate of breathing. But this can be kind of overridden, can be changed, things like that. Okay, so we see kind of see two different portions of the medulla here. So there's the ventral respiratory group here, and then the dorsal respiratory group here. Both of those are going to then communicate back either to the phrenic nerve origination site here, or these intercostal nerve origination sites down there. Okay. Yeah, and so we can see both of those. I don't know why it's shaking so bad right now, but yeah, they're jumping around up there. Well, it's not, there's another room up there, right? Yeah, yeah, but yeah, so the ventral respiratory group and the dorsal respiratory group are both involved in respiration, but not necessarily both required, but they both make their way through that, this pre-Botzinger complex area. Okay, so let's just kind of talk about which parts of the brain and which parts of the spinal cord are helping us kind of control breathing. Okay, so we're going to talk about the chemoreceptor reflex and how this works next. And so chemoreceptors, just like we talked about them controlling the heart, are also going to be important for controlling breathing. And we got them in the same two places we talked about before, right? So we're talking about well, first of all, we have central receptors actually in the medulla, sensing the level of certain chemicals in the cerebral spinal fluid. We'll talk about how those work here in a second. And then we have peripheral receptors, either in the carotid bodies or the aortic arch, right? Just those same locations we talked about, chemoreceptor and baroreceptor reflexes before. And so those are going to be sensing in the blood, obviously. So CSF, if we're talking about the central receptors, cerebral spinal fluid, and then blood here in the carotid bodies and aortic arch. Okay, so the strongest influencer of this chemoreceptor reflex is CO2, but also receptors there for hydrogen and oxygen levels. Okay, and so these are similar to what you might think. Things that are associated with increased metabolism are going to make us breathe more. Things that are associated with decreased metabolism are going to make us breathe less, right? So we got high CO2, high hydrogen, and low oxygen are all going to lead to an increase in ventilation. And then it's just the opposite for the decrease in ventilation. Okay, these are driven by these type 2 glomus cells. Okay, and those types 2 glomus cells are connected directly to afferent neurons. They're going to make their way back up to the, like you can see there, the ventral respiratory group and the dorsal respiratory group to kind of drive this reflex. Okay, so we'll talk a little bit about how we activate that type 1 glomus cell. Okay, so you can see in that little image, the type 2 glomus cell is kind of more of like a support cell that kind of holds these together, whereas the type 1 glomus cell is the actual one that's going to have the chemoreceptor property to it. Okay, so we can see this type 1 glomus cell is kind of they're right next to blood vessels. Okay, and so there's multiple mechanisms, but don't have it fully mapped out for how it senses CO2 and hydrogen, but they do have it mapped out for how it senses oxygen. Okay, so in situations of hypoxia, you have to write really slow with my pencil right now. I don't know why. So hypoxia, decreased oxygen levels, right? That that somehow affects a membrane protein here on these type 1 glomus cells that is going to lead to decreased potassium efflux. Okay, so still not even all of this mechanism is mapped out, but somehow there's a membrane protein here that's sensitive to the amount of oxygen in the capillary next to these glomus cells, and that triggers that membrane protein to then pretty much close up some of those potassium channels that are allowing for potassium efflux. Okay, and that is going to then allow for depolarization of those type 1 gamma cells. And then that depolarization is going to open calcium channels. Okay, and these are the type L or L type channels, just like we talked about before. Okay, and then we're going to get calcium influx. And then eventually that depolarization that's caused by that is going to lead to dopamine release. And that's what we see being released here. So just like the action potential going down a motor neuron causing calcium influx, leading to acetylcholine release, it's the same kind of idea here, right? We're getting depolarization because we're stopping potassium efflux. That leads to opening of calcium channels. That calcium then goes in, causes the vesicles full of dopamine to go to the surface, release, and then that's going to cause an EPSP here on these afferent neurons. Okay, questions on any of that? Go ahead. Yeah, I should have put the L first, but the L-type calcium channels, just like we had in the heart, right? Those long-lasting calcium channels. Yeah, long-acting. Okay, other questions on any of that? So that's one established mechanism of how these chemoreceptors work. Okay, and then when we're talking about central chemoreceptors, this would be happening up in the medulla, right? So now not talking about the carotid body or aortic arch, talking about way up in the medulla, sensing the level of these chemicals in the cerebral spinal fluid. Okay, even though we said it's primarily driven by CO2, we're going to see how it's a little bit more complicated than that. So when we have high CO2 levels in the blood passing through capillaries of the brain, CO2 can actually cross the blood-brain barrier. Okay, so first step is CO2 diffuses into the brain. All right, so we can see that happening here. Okay, and then it's going to combine with water, right? Drive that same reaction we saw before, right? And then it's going to eventually create hydrogen ion and bicarb. And then it's actually the hydrogen ion that binds to chemoreceptors. Okay, so CO2 diffuses into the cerebral spinal fluid, goes through that same reaction we talked about before to create carbonic acid. Carbonic acid dissociates into hydrogen and bicarbonate, and then it's the hydrogen ions that actually bind to the chemoreceptors in the medulla and kick off the chemoreceptor reflex when we're talking about those central chemoreceptors. Okay, questions on that? Okay. All right. So then let's talk a little bit about a couple other reflexive actions related to ventilation, non-chemical ones. So we have different receptors that are helping to drive inspiration, either starting inspiration or ending inspiration, and same for expiration. Okay. So we have a group of reflexes known as the Herring-Brewer reflexes. Herring-Brewer reflexes both are related to inflation and deflation of the lungs. So the inflation reflex is increased duration of expiration produced by lung inflation. Okay, so the lungs are inflating. We have stretch receptors that are being activated as those lungs are stretching out. Okay, And then the same, and then it's just kind of the opposite here. Deflation reflex, decrease in duration of expiration produced by lung deflation. Okay, so as the lungs are deflating, those stretch receptors are then sending less of a signal. Okay, and so depending on what our need is for ventilation, do we need to breathe deeper or less deep? These stretch receptors are changing the depth of our breathing pretty much by sending the right signal. The brain knows kind of how much we need to fill up the lungs to get the right amount of air, depending on the states that we're in metabolically. And then we have these rapidly adapting receptors, which are known as irritant receptors. And so these are stimulated by chemicals like histamine, which is partially what can irritate our airways when we're sick, okay, or with allergies, okay. But then they also can be triggered by things like dust and other things like that. Okay, so activation of these in the trachea is what leads to coughing. Okay, also leads to bronchoconstriction and mucus secretion. When we activate these in the lungs, this causes us just to breathe more, hypernemia. Okay, we also have C fiber nerve endings all throughout our airways, especially down in the pulmonary capillaries. Okay, and these are stimulated by capsaicin. And this is a different type of reflex that can happen called the pulmonary chemoreflex, which can lead to a brief period of apnea. So rapid breathing, bradycardia, and hypotension is all kind of triggered by capsaicin or hyperinflation of the lungs. And then lastly, proprioceptors are involved in controlling ventilation as well. So those like Golgi tendon organs we talked about and muscle spindles we talked about are involved in driving our ventilatory response during exercise. And so when our joints are moving, our muscles are lengthening and contracting, that actually plays into the dorsal and ventral respiratory group and the signal that ends up being sent out to lead to ventilation. So that's a little bit of the less understood portion of how ventilation is controlled, but it has been shown in like spinal cord injury patients, they don't have the same drive, they don't have the same increase in ventilation with exercise. So like, for example, if they don't have use of their legs anymore, but they're exercising with their arms, they don't necessarily have the same exact drive for increased breathing as people that have a fully intact spinal cord. And so that's kind of how they started realizing that some of these proprioceptors are involved in that response. Okay, yeah. It is, yeah. Yeah, it's kind of this weird reflex that I think it can be used sometimes in diagnosis of some things, but it leads to this weird like apnea where there's like stop breathing for a second, and then this like rapid breathing and your heart rate drops. I think there is some clinical application. I'm not remembering of that one exactly right now, that they can kind of test for certain things with that reflex, but yeah, it's kind of strange. Okay, a couple other reflexes here. So coughing and sneezing, definitely important for a couple different things here, but both are responses to irritants, and the differences are the locations of where the irritants are, and then the actions are different. Okay, so with coughing, irritations either in the trachea or the bronchi, right? Vagus and glossopharyngeal nerves are involved. The main thing with coughing is your glottis is closed when you're coughing. Okay. But then actually the air that you force up by contracting all of your abdominal muscles as part of the reflexes, it knocks your glottis open, right? And that's how you produce so much pressure with a cough, is because you're creating a bunch of pressure in your lungs with the glottis closed. And so you get enough pressure to just knock the glottis open, right? And then that can produce something like 600 miles per hour of air passing through your airways. We can obviously do this voluntarily as well as reflexively. Whereas sneezing, the glottis is open, irritation is in the nose instead of the airways. Trigeminal nerve is what drives this. And so yeah, sneezing is really just trying to get stuff out of your nose. Coughing is trying to get stuff out of your lower airways. A couple other ones, hiccup, you guys may already know, it's just kind of this like spasm of the diaphragm, like a quick spasm of the diaphragm. You inspire first, right, before you hiccup. You've noticed that, and then the diaphragm just suddenly contracts. The yawning, we don't really understand exactly why, but one thing we know it does is it does increase venous return when we yawn. And so it increases your end diastolic volume a little bit, which gives you a little bit more stroke volume. And so that might be something related to that, but it doesn't seem to really be needed to increase oxygen levels. That was kind of what a lot of, what people generally think it's doing is maybe have low oxygen for some reason, and that's why you're taking that big deep breath. But there's also some thought to be that maybe this helps to open up alveoli that have kind of collapsed, because we got so many million of those. Some might be collapsing all the time, and that might kind of open them up. But we don't really fully understand why that happens. So yeah, Olivia. Why is it contagious? Yeah, it's a great question. I know. It totally is. Huh? I know. I have no idea, but it definitely is. Yeah, for sure. Yeah, go ahead. Yeah, good question. No, it's not random. It's definitely directly connected to your digestive tract as well. But yeah, I think it is certain irritants in the digestive tract, in the esophagus area, and kind of the connection with the diaphragm there. But I don't know like which specific ones would do it. But yeah. Yeah. Yeah, that's true. There's a lot of hiccuping in the womb. So yeah, I don't know if that's just due to all of the the fluid in the lungs at that point. Yeah, I don't know the exact cause of it, but yeah, there's probably some kind of irritant. Go ahead. What do we got? Go. Yeah.
So if you don't do go down. On the thought, I mean, my learning question as people think it's fascinating with the chronic nerve, which makes sense. I
Yeah.
don't know why the chronic nerve would be
It's
particularly stimulated above kind of the diaphragm from the GI.
yeah. It's interesting. Yeah. That's a good.
I know a person that used to get
Hmm.
hiccups after like eat really quickly.
Huh. Yeah.
So
Yeah.
now it's also babies that have small stomachs, so it's fast. It's
Yeah.
no pressure against it.
I like this. I like this train of thought. Yeah.
So hiccups, we don't, I know we said we don't really understand why they're happening, but the ultimate purpose of them is to increase oxygen.
Hiccups?
Yeah.
No, that was the yawn we were talking about here is not needed to increase oxygen.
Yeah.
Um,
Is there a purpose for hiccups?
yeah, not that I know of. Yeah.
They're not.
Yeah, they are. I know. Yeah, there's like my kids have always a couple different like Guinness Book of World Records we read sometimes, and there's like the people that have had hiccups for like 15 years and stuff like that, like the longest like running hiccups. Like I know, right? Yeah. So, but you guys should look that up. We need to know more about hiccups. I think everybody's interested. So let's all look it up. All right? Right. Yeah, you're good. Yeah, you're good. You just had a big yawn because you were just thinking about all this and you stretched everything out. Yeah. Okay. I know. Always all these weird human things that are interesting for sure. But okay. Well, let's talk a little bit. We'll kind of finish with talking about exercise ventilation and how we drive our ventilation up so drastically with exercise. Okay. This is by far the thing that makes us ventilate more than anything else, right? 10 to 20-fold increase in our ventilation with exercise. Okay, there's multiple factors that cause this. Definitely number one would be increased CO2. And we have chemoreceptors around our muscle that are doing this. So we don't have to wait till that excess CO2 gets back up to our carotid bodies or aortic arch or CSF in our brain. It's right there at the muscle. There's being produced. Okay. We also have some anticipatory response as well. So like even as you're thinking about exercise, as you're going to put your shoes on, kind of getting ready, you're already ventilating at a little bit higher rate. So there's kind of that portion. And then the proprioceptors that we already talked about. Okay. CO2 is definitely the primary driver of breathing. And that's why we can see this relationship right here, right? The relationship between ventilation, which is also known as respiratory minute volume, and alveolar PCO2 is somewhat linear, but you do see kind of these breakpoints in the relationship as well, right? It's not a perfectly straight line. It gets a little steeper right here. It gets a little bit more steep right there. So there's those two breakpoints that we'll talk about a little bit more here in a sec too. Okay, so where is CO2 coming from? Why do we increase CO2 during exercise? Right, this slide kind of shows us that, right? We've already obviously talked about this in terms of metabolism, right? We see CO2 being released from the Krebs cycle, from this step between pyruvate and acetyl-CoA. Right, those are our primary places that CO2 comes from in our oxidative system. Okay, but then also in our glycolytic system, we're making this lactic acid, right? That lactic acid is going to donate a hydrogen ion to our solution, right? Which is then going to push this equation back this way, and we're going to get some CO2 that way as well, right? So those would be our two primary places or primary reasons we're releasing extra CO2 during exercise, is because of the oxidative system as well as buffering of acid produced by the glycolytic system. Okay, questions on that at all? Good. Okay. Well, there's still people writing, so I was waiting for that. Yeah. Nobody's that interested in exercise, I guess. It's hiccups we care about today. But yeah. Okay. Exercise sounds too hard anyways. So yeah. Okay. Okay, so a little bit more about exercise ventilation. This is kind of the acute response to exercise, right? So if we're have somebody with where we're able to measure their oxygen consumption with this device like this is called a metabolic cart, right? Where we would be measuring oxygen consumption and CO2 production. If we have a graph like this, where our O2 consumption's here on the y-axis, x-axis is just time. Okay, what we're looking at here is they start exercising at a given intensity. We kind of know that they need to consume a certain amount of oxygen to maintain that exercise intensity. But as you guys all know, we can't get our oxygen consumption immediately up to that required amount, right? We start kind of slowly, right? As we start running or whatever, or we're not already breathing as hard as we're going to. It takes about 2 to 3 minutes to get up to what's known as steady state oxygen consumption. Okay, so the first 2 to 3 minutes, we're having to use our other two energy systems to a greater degree, right? So we're using that creatine phosphate system we talked about, using the glycolytic system we talked about. And so during that time, we create something called the O2 deficit. And that's pretty much the area under the curve of that little triangle-ish thing on the left there, right? If we were able to get our oxygen right up to where we needed it, then we would have no deficit, right? But since it's kind of this gradual thing, then it's all that oxygen that we didn't consume that we should have if we were going to be using oxygen only to drive exercise. Okay, so then after about 2 to 3 minutes, the aerobic system kind of catches up, because that's pretty much what causes this. Is our oxidative system has so many steps, right? It takes a long time to get all the way from glucose to the electron transport chain, make all that ATP. So it just takes a while, right? And so it takes a couple minutes to get everything going. But then once we do, then we get to steady state oxygen consumption. Okay, and then the other end of this is when all of a sudden we just stop exercising. We all know that our oxygen consumption doesn't immediately go down to where it started, right? It takes a while to get down there. And that area under the curve here is known as EPOC, which is just this acronym, excess post-exercise oxygen consumption. And this, it used to be thought that the O2 deficits in EPOC were just kind of like the exact same, right? Like EPOC, the cause of EPOC, initially what people thought was that it was just repaying what wasn't, it's kind of like a debt. They used to call this the O2 debt, because then they thought we were just repaying all of it right here. But it tends to be that EPOC is quite a bit bigger. And so some of the things are kind of repaying the debt. So we're replenishing oxygen stores. Okay, so if we used up some of our stored oxygen in our muscles and our blood, we're going to replenish that. That's some of what we're seeing. We're replenishing our creatine phosphate. To put that phosphate back onto creatine requires some energy, requires some ATP to do that. So it's going to be part of it. We're going to have to deal with lactate. We talked about what happens to lactate before. Most of our lactate is burnt via our oxidative system just by converting it back to pyruvate. And so that's that requires some oxygen if we're burning that lactate via the oxidative system. And then also just things like an increased temperature. When our temperature is higher, we're less efficient and we use more oxygen. Okay, so that's going to be part of it. Also, high levels of epinephrine in the blood drive up our metabolism too. So we're going to have kind of a, when we're exercising, we release a lot of epinephrine, and then it's going to take a while to clear those levels out. So all these things are kind of causing this epoch situation where it takes a while to get respiration back to normal. Yeah. O2 deficit would be, when you just start exercising, it's the amount of oxygen that you didn't consume that you would have if you were able to just fully use your oxidative system immediately to the level you got to, right? So because it takes a little while to get your oxygen up to the level that it needs to be to sustain whatever exercise intensity you chose. And so, it's that oxygen that's not consumed, but it would have been consumed if you were able to fully use the oxidative system for every second of that exercise. Okay. Other questions on that? Yeah. Go ahead.
So, like back when everybody was doing all of like the HIIT stuff, where
Uh-huh.
you would do like 2 to 3 minutes of exercise, and then you wouldn't really let yourself recover,
Mhm.
like recover for 30 seconds, and then you do another 2 to 3 minutes.
Yeah.
How does this graph like look different for somebody who's trying to train their like anaerobic system like that, where they're trying to increase their EPOC?
Mhm. Yeah, I mean, so pretty much anytime you change your exercise intensity, you're going to have a new O2 deficit. So let's say you start with like an easy intensity, it'll take 2 to 3 minutes to get up to steady state. And then if you go faster from there, take 2 to 3 more minutes to get up to that new steady state. So with HIIT, it's kind of like, it's going to take, you're going to have a larger O2 deficit the higher the intensity is, for one thing. So it'll take longer to get up to steady state if you're at 90% of your max, like you need to be with HIIT, right? And then that O2 will drop back down a little bit during the recovery period. And then it'll just come back up. And then it won't take as long to get back up the next time because you're already higher than resting, right? And so it'll kind of be just looking like up and then down, but it'll kind of rise continually too. Usually by the end of like a 4 interval HIIT or a 10 interval HIIT, we'll see the heart rate kind of slowly going up higher and higher and higher, just because you have so much more sympathetic activity as you get more and more fatigued. But yeah, I don't know if that kind of answers your question, but yeah, exactly. Kind of like steps. Yeah. Other questions on that? And this EPOC thing is something that people are interested in for weight loss, because it's not just during exercise that you're burning the calories, but the harder you work out during the workout, then the larger EPOC you have, right? And so it's known as like afterburn, if you've seen that too. Doesn't probably have a huge effect on weight loss, but it is something that is touted to be helpful for burning a few extra calories after the workout. So, yeah. Okay, so a couple more things here. The thing I was noting back here, where we had these kind of break points in that ventilation to CO2 relationship, is all related to these factors known as different threshold points in ventilation. Okay, so when the ventilation threshold is known as the point at which, so normally, so this would be an exercise test where the exercise is getting slowly harder and harder and harder. Like maybe you're on a treadmill, walking at a certain speed, and maybe every minute the grade goes up by 1%, something like that, right? Like it's getting just a little bit harder each step of the way. So during that time period, your ventilation is going up every time that exercise gets harder, right? And that's initially here in the first part down here, that's pretty much all driven by the extra CO2 produced by using your oxidative system, right? As we're using more and more of our oxidative system, we're making more and more CO2. That's going to be making us breathe a little bit more, okay? But then at some point right around here, as you can see here, there's this threshold point to where all of a sudden it takes a steeper linear path, right? It gets steeper right there. And that tends to somewhat line up with the same point at which our lactate starts accumulating in our blood, right? So lactate threshold is when lactate starts accumulating in the blood. ventilation threshold is when that ventilation gets steeper, but they tend to be somewhere around the same spot. Okay. And so the reason for that is because now we're not just getting more CO2 from the oxidative system, but now all of a sudden lactate's accumulated in the blood. So now we need to start activating that buffer system. And so that's producing more CO2, right? So we get more CO2 from the buffering system there, right? And so now we've got the extra going from the point before the threshold to a point after the threshold. We've still got more CO2 coming from the oxidative system because we used our oxidative system more, but now we've also got that added CO2 coming from buffering as well. And so that's making that this new linear relationship where it's a little bit steeper now than it was previously. Okay, so so we can kind of walk that's the threshold part, but we can kind of walk through these steps. What's causing ventilation to increase immediately? This is going to be mostly driven by psychological factors. This is like when we first start exercising. And then this is where those proprioceptors are really important as well. Because we don't immediately produce a bunch of extra CO2. It takes a while for that to get back into circulation. So our immediate two things are really driven by psychological factors and proprioceptors. As your joints are moving, that's sending the signal to the brain, okay? Then once we get kind of up to the moderate exercise level, where we're not just starting, but we're kind of breathing somewhat hard, that's going to be mostly driven by CO2. This thing is killing me today. I really need to get a new one. CO2 from oxidative metabolism, as well as some heat production. As our body starts warming up, that's also part of the drive to breathe. And then we also have an increase in potassium levels in the blood as we're doing so many action potentials on our muscles and on our nerves. We're constantly sending potassium back out to repolarize and hyperpolarize, right? And that eventually just starts building up some potassium in our extracellular space. That stimulates some chemoreceptors for this as well, okay? But then when we get all the way up into kind of real higher level exercise, now again, we've got CO2 from the oxidative system, but also CO2 from buffering. And those are kind of the two primary drivers to get us up further to this kind of real high level of ventilation that we can only get with really intense exercise. Okay, questions on that at all? Okay, and then lastly, this our ventilatory system adapts to exercise as well, right? And we don't have to breathe as hard at a given intensity after we've trained for a while. And so these graphs are just kind of classic graphs that talk about this relationship a little bit. So if we're looking at somebody who's untrained versus somebody who's a trained runner, we can see a couple of things here. So this is our percentage of VO2 max, or like percentage of our maximal capacity to exercise that we're at. Okay, and so we can see pH there, we can see ventilation, right? So two different measurements. And so, yeah, maybe just for a second with the people next to you, discuss what are the differences that you see between the trained person and the untrained person as far as their ventilatory response to exercise and how that might relate to their pH in their blood.
It goes down to the same age, but the untrained student. Like, oh, your pen, and I was like.
All right, let's go ahead and come back together here, finish this up. Okay, so what are some differences, or what's a difference somebody noticed? Somebody raise their hand and tell me. What do we got? Go ahead. Have a sharp drop in which one? More gradual. Yeah, we do kind of see this more gradual relationship. And then this is kind of a steep drop here. Okay, what else? What else we say? Go ahead, Quinn. Yeah, can get to a higher percentage of their VO2 max with less ventilation. Definitely as far as the threshold concept, we're seeing that ventilatory threshold here as well. So this would kind of be the ventilatory threshold in the trained one. Whereas here, we're looking at it probably somewhere like in here instead. Right? And so I think this is kind of what you're getting at, Quinn, with we see ventilatory threshold happening much later, or at least into a higher percentage of the trained person's VO2 max. So maybe only around 50% in the untrained person, then up around 75% in the trained person. Okay, so we're able to kind of breathe at a more moderate rate when we're trained, up into a higher percentage of our VO2 max versus when we're untrained. We're going to start breathing heavy earlier on. And this isn't even like an absolute VO2 number. This is percentage of VO2 max, right? So VO2 max definitely is higher in the trained person as well, but this is just, they can actually get to a higher percentage of their VO2 max with a lower ventilation as well. And then how's this related to pH? They're pretty much right next to each other, right? We see the drop in pH kind of really taking effect around the same time that the increase in ventilation happens, right? So we're seeing, we're pretty much seeing that ventilatory. First of all, we can say VT for that ventilation threshold goes up with training. Okay, pH stays up longer with training as well, right? We can kind of maintain a higher pH longer after training. Okay, and the biggest reasons for that are the training improves our oxidative capacity, which means less reliance on that glycolytic system. All right, so this is really more of an adaptation of our muscle cells and our ability to deliver oxygen to the muscle cells than it is the lungs itself, right? The lungs are kind of just responding to whatever we need them to respond to. But when our oxidative system becomes stronger, then we can use our mitochondria more. We don't up into higher intensities because the mitochondria can keep up with the demand for oxygen or the demand for ATP. And so then that means we don't have to kick in this glycolytic system as early, right? So we don't produce lactate as early. We don't have to buffer that extra CO2 as early. So we don't get that steep rise off of this linear relationship until later on. And really has a lot mostly to do with the energy system adaptations that happen with exercise. So, okay, questions on any of that? All right. Well, we're done 22 minutes early. Have a wonderful Thanksgiving break, you guys. Hope you get some rest. We'll see you next week.
Yeah, what? What is? That's fine.